So before we move into our section on antiviral agents, I want to take a brief detour to discuss one of the first approved gene therapy treatments known as Casgevy. Casgevy is a CRISPR-based gene therapy designed to target sickle cell disease. So the major goals for this lecture are to review the molecular basis of sickle cell disease, understand fetal hemoglobin regulation, explain how CRISPR-Cas9 is used in Casgevy, compare gene editing therapies with small molecule drugs, and discuss the benefits, risks, and limitations of CRISPR-based treatment. I would like to first acknowledge three of my recent biochemistry students, Destiny Lopez, Krista Haas, and Annie Ford. They put together a really nice presentation on CRISPR gene therapy techniques as part of a project for their Chemistry 450 Biochemistry 1 course. I will be using a lot of the content that they put together for their presentation. So I wanted to acknowledge them and thank them for their hard work on the project and for their contributions to this lecture. First, I'd like to review sickle cell disease. Sickle cell disease most commonly results from a single amino acid substitution in the beta globin subunit of hemoglobin. Recall that each person has two alleles or two copies of each gene, one that they receive from their mother and one that they receive from their father. In the case of sickle cell anemia, if the two parents each carry the sickle cell trait, i.e. they have one wild type and one diseased allele, they can pass these traits on to their children in one of four ways. This gives a phenotypic ratio where 1/4 of their children would have both wild-type copies of the gene, half of their children would carry one wild-type and one mutant allele. This is the optimal condition for protection against malarial infections, and 1/4 of the children would have both mutant copies of the hemoglobin, giving these children sickle cell disease. So in our lectures on malaria, we discussed how having a single sickle cell allele was a protective advantage that reduced malarial infections. However, if people have both sickle cell alleles, they develop sickle cell anemia, which is incredibly painful and a life-threatening disease. The mutant hemoglobin has a sticky patch on it that causes the hemoglobin to stick together, especially when it's not actively carrying oxygen. This forms long tangles of hemoglobin within the cells and distorts their shape into the sickle cell. The sickled cells can then aggregate together in small capillaries within the bloodstream and block blood flow in that region. This can cause tissue and organ damage. Major risks over time include fatal organ damage to the kidneys, liver, heart, and lungs, high infection susceptibility, stroke, and anemia. These complications frequently lead to hospitalization, reduced life expectancy, and significant physical limitations. So let's get into the technology that is enabling gene therapy. So similar to all other living organisms, bacteria can also be infected and suffer from viral infections. Here you see a bacteriophage viral particle attacking a bacterial cell. The viral genome is then injected into the bacterial cell where it will be expressed and take over the metabolic system of the bacteria for the production of more viral particles. It generally kills the bacterial cell in the process. Thus, bacteria have developed a unique immune system response to viral infections that we are now exploiting for human gene editing purposes. It's really quite remarkable. So how does this system work inside the bacteria? CRISPR stands for clustered, regularly interspaced, short palindromic repeats. That's a mouthful for sure. These are unusual repeat sequences in E. coli, shown here. They're separated by non-repeating sequences called spacers. These are shown here in this diagram where you have the repeat palindromic sequences interspersed by the spacer regions. Palindromic sequences of DNA are sequences that are the same going in the forward and the reverse directions. So recall that DNA is a double helix made up of two strands of DNA. They're held together by hydrogen bonding between the DNA bases, A with T and G with C. These are also directional molecules that lie head to tail with one another, moving in either the 5' to 3' direction, and the other in the opposing 3' to 5' direction. So here you can see the palindromic repeat sequences interspersed with the spacer regions. The palindrome is shown here in the red sequence. You can see that the 5' to 3' sequence on this top strand reads C, G, T, T, G, A, G, and so on. If you look at this strand down here and follow it in the 5' to 3' direction, you will see the same DNA sequence. C, G, T, T, G, A, G, and so on. This is what is meant by palindromic repeats within this region. These palindromic repeat spacer regions are found in about 40% of all eubacterial species and 90% of the archaebacterial domain. The spacer regions are derived from foreign chromosomal DNA, most often from bacteriophages. Thus, when a bacterium is infected by a virus, this adaptive immune response is activated. This involves a Cas nuclease, which is an enzyme that can cut DNA or RNA. The Cas nuclease recognizes the foreign DNA and cleaves it into these bite-sized fragments. The fragments of the foreign DNA are then integrated into the CRISPR repeat sequences as the spacer regions that are shown here. Once the CRISPR system is built, this provides the adaptive immunity to the bacterium. If the bacteria is then exposed to more of that same virus or to similar viruses, the CRISPR-Cas response system is activated. This will cause the production of a Cas nuclease enzyme, Cas9 in this case, and a trans-activating CRISPR RNA sequence called tracer RNA. This also causes the transcription of the CRISPR repeat spacer region, producing a pre-CRISPR RNA molecule. The Cas9 nuclease will assemble with the tracer RNA, shown here. It will bind with the repeat sequences in the pre-CRISPR RNA molecule, and an additional RNase enzyme will cleave the pre-cRNA into segments. Each unit is then fully assembled to function in the immune response. The assembled CRISPR-Cas unit can then bind to new foreign DNA, shown in green here, that may be entering the cell. The Cas9 RNA complex randomly interrogates DNA in the cell, searching first for the appropriate proto-spacer adjacent motif. This is the PAM motif. It's a short motif that lies adjacent to the target spacer sequence. Upon recognition of the PAM sequence, the Cas9 RNA complex unwinds the DNA or RNA from the first 10 to 12 nucleotides following the PAM sequence. This is termed the seed region. If the interrogated DNA sequence matches the CRISPR RNA spacer region, the nuclease of Cas9 will cleave both the non-target strand and the target strand. This causes a double-stranded break in the foreign nucleic acid, whether it's DNA or RNA. Thus, the bacteria can evade the infection and become healthy again. So we've further adapted this system for use in gene editing purposes in humans and other species. So how does this work? In 2020, Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize in Chemistry for their work with CRISPR-Cas systems. Doudna and Charpentier showed the first use of CRISPR-Cas9 to introduce double-strand breaks in target DNA. This involved the use of a CRISPR-Cas assembly, as shown on the left. This required two fragments of RNA, the tracer RNA and the guide RNA, that housed a homologous binding region for the tracer RNA attached to the target sequence for the target gene where the strand break should be introduced. Following this initial experimentation, they further modified their system to become more reliable by linking the two strands of RNA together with a small extra sequence of RNA, as shown here on the right. Once the CRISPR-Cas system has made the DNA strand breaks at the appropriate location, the cell's natural repair mechanisms, either non-homologous end joining or homology-directed repair, can be utilized to repair the breaks and restore the DNA. Ultimately, this results in either a deletion or insertion into the region and can disrupt the open reading frame of the gene sequence, creating a knockout. Here's the process of non-homologous end joining and homology-directed repair in a little more detail. HDR requires homologous regions of the DNA that will help align and lead to the repair of the damaged region, and it's shown here on the right. If no homologous overhangs are present or are very small, the DNA is repaired through the non-homologous end joining pathway. This can generate either deletions or insertions during the repair process. Alternatively, a donor DNA can be provided in trans that will lead to a transgenic DNA that can be created, essentially repairing or replacing a damaged portion of an open reading frame. This later process still poses many challenges to being used in patients. Gene editing is not without risks. Even though it's highly specific for a certain location, there are possible on-target side effects. These include chromosomal abnormalities or other smaller gene modifications that are unpredicted. And furthermore, in the case of sickle cell, if you actually repair both sickle cell alleles, you can also increase a person's susceptibility to malarial infection if they live in a region with high infection rates. Gene editing can also cause off-target effects. This could involve gene editing away from the desired location or off-target, and can lead to the creation of other defective cells and increased risk for deleterious effects that could make the patient even sicker. Off-target effects could have a drastic consequence. For instance, they might change DNA sequences that control cell growth, making cells cancerous. Other unwanted changes might prevent cells from carrying out important functions. For example, they might prevent the pancreas cells from producing insulin. So how does Casgevy work as a gene therapy tool to cure sickle cell disease? It may surprise you to learn that this gene editing does not seek to repair the mutation in the beta hemoglobin gene. Making repairs to genes is still a harder process to achieve than it is to deactivate or disrupt gene function. Instead, Casgevy targets a gene called BCL11A for disruption. To understand what the gene product of BCL11A is doing, we must first understand a little bit more about the types of hemoglobin proteins present inside the body. There are multiple forms of hemoglobin present inside the body, and we're going to discuss 3 of them. We've learned previously about the alpha and the beta modules that make up the common tetramer of hemoglobin protein inside humans. In the sickle cell conditions, recall that a single point mutation occurs in the beta subunit and converts a glutamic acid residue to a valine residue. The hemoglobin tetramer with the alpha and beta subunits is shown here, with the beta subunit shown in yellow, the heme cofactors shown in red, and the alpha subunit shown in light red. There's also a gamma subunit of hemoglobin that is expressed instead of the beta subunit within a developing fetus. This is shown in the light orange in this tetramer of hemoglobin. So you've got 2 gamma subunits and 2 alpha subunits. This fetal form of hemoglobin has higher affinity for oxygen, allowing the developing fetus to take the oxygen from the mother's blood more easily and ensure that the growing baby has enough oxygen for development. This is shown in the graph to the right that shows the differential oxygen binding of the adult hemoglobin compared with the fetal hemoglobin. Around the time of birth, the fetal A gamma and G gamma globin expression is silenced, and the adult beta globin gene is turned on. This process is referred to as gamma to beta hemoglobin switching. Notably, the zinc finger transcription factor BCL11A has a major role in silencing the gamma globin expression in human cells, so it turns this off. Thus, the strategy of Casgevy and other sickle cell gene editing pharmaceutical agents is to disrupt the expression of the BCL11A. In the absence of BCL11A, the fetal form of hemoglobin can be expressed. It can then replace the mutant beta hemoglobin subunit within the hemoglobin tetramer. So, what does this do physically in the patient? Inhibiting BCL11A does not inhibit beta hemoglobin production. Instead, it reactivates gamma globin expression. So the red blood cell produces a mixture of hemoglobins, with the fetal hemoglobin, HbF, becoming dominant. Now the cell produces HbF with 2 alpha and 2 gamma subunits. It can also still produce the HbS, the sickle cell version, with the 2 alpha subunits and the 2 beta subunits that have the mutation. Or it can produce a mixture with 2 alpha, 1 beta, and 1 gamma subunit. So the beta HB is still present, but it's diluted, and it's more like having only a single allele of the sickle cell trait. So why does this work therapeutically? HbF does not participate in sickling, and in fact, it interferes with the HbS polymerization and red blood cell sickling. Even partial expression of the fetal hemoglobin dramatically reduces sickling. This is also a safer and more effective target as the expression is being disrupted of the BCL11A protein rather than the beta hemoglobin gene being repaired. Thus, higher efficacy can be achieved in this process. So what happens to the patient during treatment? Different treatments can work in different ways. Casgevy follows this path. It uses ex vivo gene therapy that relies upon the isolation of patients' hematopoietic stem cells, HSPCs, from their bone marrow. Within a test tube, these genes are manipulated using the CRISPR-Cas gene therapy system. In the case of Casgevy, this involves the inactivation of the BCL11A gene, which in turn reactivates the expression of the fetal hemoglobin. Other strategies could also include the ex vivo gene addition based on a virus-mediated delivery of a functional copy of the hemoglobin beta gene, or the ex vivo genome editing of the beta hemoglobin mutation, which is involved in the nuclease-based HDR-mediated correction of the HBB gene. This is again harder to do. So before the modified cells can be infused back into the patient actually needs to be conditioned. So what does that mean? Myoablative conditioning for sickle cell gene therapy is a high-dose chemotherapy process, typically using busulfan, which is designed to destroy existing bone marrow stem cells. This allows the modified cells to engraft into the bone marrow of the patient. This is an essential step, it's a rigorous step, and it enables a cure for the sickle cell disease. But it carries risks like infertility and infection and severe toxicity. There's major, potentially life-altering side effects that include this infertility, long-term organ damage, hair loss, mouth ulcers, and high susceptibility to infections. So this process is quite painful for patients and has high risk associated with it. But so does having the sickle cell disease. So due to its ability to cure sickle cell, it is an attractive option to people that have sickle cell disease. However, due to this high toxicity, researchers are investigating non-genotoxic, fertility-preserving, or reduced intensity conditioning strategies to make gene therapy accessible to more patients. They're also trying to develop novel in vivo gene therapy that would rely upon direct infusion of the editing machinery into the body to the target patient's resident hematopoietic stem cells without isolation, manipulation, conditioning, and re-infusion. Just a direct in vivo therapy. That's the dream. Not yet a reality. So is Casgevy a drug? It's not a small molecule. It's only given as a one-time intervention, and it alters gene regulation and expression. So Casgevy really challenges the traditional definition of a drug. Rather than repeatedly administering a chemical compound, this therapy permanently alters gene regulation through a one-time genetic intervention. Some key differences between traditional drug molecules and gene therapy include the fact that gene editing is permanent. It also has high upfront risk. Small molecules, on the other hand, are reversible. They're metabolized and cleared from the body. They can also be used repeatedly. However, unlike antibiotics, antimalarials, or other small molecule therapies, CRISPR-based therapies are not subject to classical resistance mechanisms. However, they do introduce new challenges related to safety, ethics, and long-term monitoring, including these off-target sites that could be affected through the gene therapy process and that are unpredictable. Casgevy raises important ethical questions about who benefits from advanced therapies, how they're distributed globally, and where society draws boundaries around gene editing. So why does Casgevy matter to medicinal chemistry? Casgevy represents a shift in how we think about therapeutics. While it's not a traditional small molecule drug, it's still grounded in target specificity, mechanism of action, and risk-benefit analysis. These are all the core principles of medicinal chemistry. So key takeaways from today. Casgevy demonstrates how a deep understanding of molecular biology can lead to revolutionary treatments. As medicinal chemistry evolves, future therapies may increasingly involve gene regulation rather than enzyme inhibition alone.